Fluid Mosaic Model. The arrangement of phospholipids and proteins in biological membranes is described by the Fluid Mosaic Model presented by S. J. Singer and G. Nicholson in 1972. The biological membranes can be considered as a two-dimensional liquid where all lipid and protein molecules diffuse more or less easily. The cell membrane consists primarily of thin layer of amphipathic molecules having both hydrophobic water-hating region and hydrophilic water-loving regions, phospholipids which spontaneously arrange so that the hydrophobic tail regions are shielded from the surrounding polar fluid, causing the more hydrophilic head regions to associate with the cytosolic and extracellular phases of the resulting bilayer. The arrangement of hydrophilic heads and hydrophobic tails of the lipid bilayer prevent polar solutes, example, amino acids, nucleic acids, proteins, carbohydrates, and ions from diffusing across the membrane, but generally allows for the passive diffusion of hydrophobic molecules. Associated with each membrane is a set of membrane proteins that enables the membrane to carry out its distinctive activities. Membrane proteins can be classified in two broad categories: integral, that is intrinsic; peripheral, that is extrinsic. Integral membrane proteins have one or more segments that are embedded in phospholipid bilayer, having one or more covalently attached fatty acid chains that help them anchor to the membrane. Peripheral membrane proteins do not interact with the hydrophobic core of the phospholipid bilayer. Instead, they are usually bound to the membrane indirectly by interactions with integral membrane proteins. These proteins interact with membranes in different ways on the cytoplasmic side of the membrane. Some membrane protein connect to the cytoskeleton. On the exterior side of the membrane, some membrane proteins attach to the fibers of the extracellular matrix.
For more such videos on all subjects, practice questions, and a lot more, download iPrep and learn unlimited.